Good morning, student. Today we are going to start with the lesson number 6, that is photosynthesis. It is a part of plant physiology. First, we had studied absorption of water, second, transpiration. Now, this is the third part of plant physiology, that is photosynthesis. It's the most significant life process which provides food not only for the plant but to all the animals, which includes human as well. Also, it provides the life supporting free oxygen gas in the atmosphere. Now, a series of experiments on the photosynthesis, they are worth performing, and I hope you will like this lesson and this presentation. So let's begin. These are the few important points from our scope of syllabus, which we are going to talk about and or discuss about the same throughout this presentation. We'll talk about the process, definition, significance of photosynthesis, site of photosynthesis, that is chloroplast, its internal structure, how the stomata opens and closes, what is the balance equation of photosynthesis, two important steps in the light reaction as well as dark reaction, correct? What are the different adaptations the plants go for photosynthesis purpose, different experiments, and last but not the least, the diagrammatic representation of carbon cycle. Definition: photosynthesis is defined as it is a process by which living plant cells containing chlorophyll produces food substances, that is glucose and starch, from carbon dioxide and water using light energy. Why it is so important? As I told you earlier, that photosynthesis is required because it produces food for all. The photosynthesis is ultimate source of energy and the food for all living things, being directly for plants and indirectly for the animals. Second, the oxygen which is produced from this process, which is the only biological process right now available for the human beings. No living beings can remain alive without oxygen. So hence, the photosynthesis is really a very important process. Site of photosynthesis, which is known as chloroplast, contains a vital plant pigment which is called as chlorophyll. Chloroplast, they are minute double membrane structure. The shape is oval, as you can see in this picture. It's oval shaped bodies containing double membrane. This is outer membrane. This is inner membrane. This is outer membrane. Inside the chloroplast, we can see some structures. We'll come back to that structures later on. First, let's see the location. It is located mainly in the mesophyll cells. The meaning of the word meso is middle, phyll is the leaf. The cells which are present in the middle of the leaf are called as mesophyll cell, and these mesophyll cell contains chloroplast. Number, it will vary from 40 to 50 per cell, and in terms of surface area covered by the chloroplast on the leaf, we can say that 5 lakh chloroplasts are present per square millimeter of the leaf surface. So you can see that it is covering the very large amount of area, and hence the amount of food produced and oxygen release is maximum. If you look at the internal structure of chloroplast, it contains two important points. Number one, these structures, which we call them as thylakoids. Remember the individual part, this 1, 2, 3, 4, 5, 6, all these individual parts, student, they are called as thylakoids. Correct? When you start placing one thylakoid, one on the above, and you are making a pile of it or a stack of it, then we call that one structure as a granum. We called it as one structure as a granum. So 1, 2, 3, 4, 5, 6, 7, 8, 9. So I can see that there are multiple granum present in the chloroplast. I will use that word grana for it. Granum is a singular word, grana is the plural. Each granum, it is connected by a line or a structure that is called as freight. That is called as freight, or it is also called as stroma lamellae. Now, These grana, they are embedded in a ground substance, and this ground substance is called as stroma. You can compare the stroma with the cytoplasm present in the side of the cell. The wall of the thylakoids contain a pigment. That pigment is called as chlorophyll. There are 9 identified types of chlorophyll, but best known are only 2, chlorophyll A and chlorophyll B. All the types of chlorophyll, they contain carbon, hydrogen, oxygen, nitrogen and magnesium. Chlorophyll, they will absorb blue and red color light spectrum from the visible light or sunlight and reflects green. So hence the maximum rate of photosynthesis can be observed or studied during availability of blue and red light spectrum. Naturally, which is not possible. So hence chlorophyll absorbs only blue and red light spectrum from the visible light and reflects the green. It doesn't mean that if the intensity of light increases, chlorophyll will activate for more time. And is the rate of photosynthesis increases? Answer is no, because too much of the light can destroy the chemical structure of chlorophyll and it can get decomposed and the entire process will be stopped. For photosynthesis, not only solar energy is required, but carbon dioxide need to be absorbed from the atmosphere. That work will be done by the structures present on the lower side of the leaf, that is called as stomata. The opening and the closing of stomata, it will be depend on how much amount of water is accumulated in the guard cell. It will be totally depend on that movement of water, whether it will move inside the guard cells or outside the guard cell. And this movement of water, it is controlled by the solute present inside the guard cell. So there are two uh, famous theories about the opening and closing of stomata. One is potassium ion concentration and second is sugar concentration theory. This diagram is given in our textbook, I guess on page number 69. There you can see that two guard cells along with some other cells. Those other cells are called as epidermal cells. These cells are going to play an important role in providing the potassium ions. Okay. The questions can be asked, sketch and label when the stomata is open and when the stomata is closed. So you can refer that diagram from page number 69. Potassium ion concentration theory, it talks about, or it can be explained or studied like this. During day, when the photosynthesis process is going on, some amount of ATP it is produced. Correct? Now this ATP, it will pump the potassium ion from the adjacent cells. And those adjacent cells, student, we call them as epidermal cells. So the potassium ions from epidermal cells, it will be pumping into the guard cells. It means ATP, it will absorb that potassium ions from epidermal cell to the guard cells. Now, as soon as inside the guard cell, potassium ion concentration increases due to more accumulation of the same, solute concentration increases. And if the solute concentration increases, water will start entering into the guard cell by the process called as endosmosis, result into increased turgidity of the guard cell and stomata opens. This will happen during daytime. Exactly reverse process will take place during night. All the potassium ion which were absorbed or entered into the guard cell leaves the guard cell, go back to their adjacent cells. Solute concentration decreases inside the guard cell, resulting into the loss of water. Turgidity decreases and stomata gets closed. Right? So this is directly, we can say that concentration of potassium ion regulating opening and closing of stomata. This is most recent theory, so it is widely accepted. Second theory is sugar concentration theory. According to this old sugar concentration theory, during daytime, the guard cells begin photosynthesis, and the sugar, that is glucose, produced during this process increases the osmotic pressure, which will draw the water from adjoining cells, due to endosmosis, and hence the guard cells become turgid and bulge outwards due to their thin outer wall, thus whitening the stomata, and stomata gets open. Closing of stomata, for any reason, the water content inside the leaf fall down. The water drawn out from the guard cells due to exosmosis, and it will make the guard cells flaccid. As a result, the inner thick wall gets straightened and stomata get closed. Process of photosynthesis will be completed in the mesophyll cell, both palisade as well as spongy mesophyll. As I told you, these cells contain more number of more number of chloroplasts. Raw material required will be carbon dioxide and H2O. You can see in this picture, these are the two types of mesophyll cell, palisade and spongy. Palisade mesophyll cell, they are very close to the epidermis, upper epidermis. Hence, they do have more number of chloroplasts. The rate of photosynthesis can be more in palisade tissue as compared to the spongy mesophyll. Because spongy mesophyll, they are busy, or they are more concerned with the process of transpiration. The overall chemical equation for the process, it will be 6CO2 plus 12H2O in the presence of light energy and chlorophyll gives out glucose, water and oxygen. This process will be completed into two main phases. One is light dependent phase, which is also called as photochemical phase, or light independent phase, which is also called as biosynthetic phase, or simply dark phase. First is light dependent phase, also known as photochemical phase, will be completed in the thylakoids of the grana. It is divided into two sub-steps. Step number one, activation of chlorophyll. This activation of chlorophyll will occur only on exposure to the light energy. As you can see in this picture, photons, which is known as smallest unit of the light energy, it will strike on the chlorophyll molecule. The chlorophyll molecule will get excited by absorbing the energy coming from the photons, and from their ground state, that is from their normal or stabilized state, they become unstable or get excited. They again come back to their ground state, but while coming or returning to the ground state, whatever energy is absorbed by them, it will be released. This energy which is released by the chlorophyll molecule returning to the ground state will be absorbed by the water molecule, and the water molecule, it will get split into the two components, hydrogen and oxygen, and electrons also released. Since this reaction takes place in the presence of light, we called it as photolysis, which means splitting of water by light, or simply splitting of water into hydrogen and oxygen in the presence of light as well as grana, because thylakoid is a part of grana. Now at the end of this photochemical phase, student, we will get three important products: NADPH, oxygen and ATP. NADPH can be formed by NADP will accept the hydrogen electrons and get converted into NADPH. Full form, it will be nicotinamide adenine dinucleotide phosphate, which is also known as hydrogen acceptor. In the presence of enzyme, it will accept the hydrogen and becomes NADPH. The oxygen, which it will be given out in the molecular oxygen form. The electrons are used in converting the ADP, that is adenosine triphosphate, into energy-rich compound called as adenosine triphosphate by adding the phosphate. By adding phosphate, which is in inorganic form. Remember, the addition of phosphate is called as phosphorylation. But actually, this reaction is taking place in the presence of light, because this electron, energy-rich electron, we have obtained from the photolysis of water, right? So This ADP, the conversion of ADP plus phosphate into ATP, it is called as photophosphorylation, addition of phosphate in the presence of light. In the presence of light. Second step is light independent phase, which is also called as biosynthetic phase. This phase was discovered by a scientist named Kelvin. Hence, you can call it as Kelvin cycle. It is also called as dark phase because it do not require light energy and it occurs simultaneously with the first phase. Please remember, it will occur simultaneously with the previous step, that is photochemical phase. It will completed into the stroma of chloroplast. NADPH and ATP, what we had studied in earlier slide, that they are produced in the grana. These two products, they will leave the grana, enters into the stroma of the chloroplast, and it will get breakdown into NADPH will get breakdown into NADP plus H, and ATP will get breakdown into ADP plus E plus electron. This hydrogen and electron, student, it will be accepted by the carbon dioxide enter from the stomata. This carbon dioxide accepts hydrogen and electron, gets converted into glucose, and glucose, since it is a very simple product, can get absorbed by the plant cell, hence it will be converted into starch. Hence it will be converted into starch. Correct? Now, this is what is the summary of these two phases, light dependent and light independent. As you can see, in the thylakoids, photolysis of water takes place due to activation of chlorophyll, produces oxygen, hydrogen and electron, right? This oxygen, it will be given out. Hydrogen, it will be converted into NADPH because it will be accepted by NADP. Electron along with ADP plus P converts into ATP by the process of photophosphorylation. So it means ATP, NADPH, they are produced in the thylakoid, that is grana, right? Now these two, NADPH and ATP, they will leave the grana, enters into the stroma. NADPH will break down into NADP plus H, hydrogen will go to the carbon dioxide, NADP again enters into the thylakoid and complete this cycle. So it goes on continuously throughout the day and the time. Similarly, ATP, it will get breakdown into ADP plus P and the electrons are released. Hydrogen and electron will be accepted by carbon dioxide forming glucose as well as starch. Got it? Now, there are certain adaptation take place in the leaf to perform the photosynthesis. Number one is larger surface area. For maximum light absorption, it is required. Narrowing of the leaves in certain plants like tulsi or basil, it will minimize the transpiration in order to conserve the water. For example, oleander or pine, or as I told you, basil. But if you see in case of banana, the surface area is more and hence the photosynthesis will be maximum. Second is leaf arrangement. Student, in biology, we called it as phyllotaxy. Arrangement of leaf on the branch or the plant. All the leaf should be at the right angle so that light source obtain maximum light, so that they can obtain maximum light from the light source, that is sun. Cuticle and upper epidermis. Remember, the cuticle which is present on the upper epidermis should be transparent or as thin as possible, but also it should make sure that it is waterproof. Water should not get lost and allows the light to enter inside the leaf freely. Maximum number of stomata allows rapid exchange of gases. Okay, so oxygen and carbon dioxide. Carbon dioxide will be absorbed and oxygen is released. Correct? So numerous stomata are required for maximum photosynthesis. Thinness of the leaf. The thinness of the leaves will reduce the distance between the cell, facilitating rapid transport or translocation of the food which is produced. Number of chloroplasts need to be concentrated near the upper epidermis because uh, those chloroplasts will obtain light energy quickly. Extensive vein system is required for rapid transport to and from the mesophyll cells, whether it is of water or food which is produced in the form of glucose or water. Now the three end products of the photosynthesis, which was glucose, water and oxygen, what will happen with them? What is the fate of these three products? Glucose, being a simple sugar, it will be used by four different ways. As you can see, it will be immediately consumed or can be stored in the form of starch, may be converted into sucrose, or it is used for the synthesis of protein or fats. The water produced in this process, it will get reutilized in the continuance of the photosynthesis or any other uh, life processes pertaining to the plant body. Oxygen, some of the oxygen can be used by the plants by the process of respiration, but major portion of it, it is not required and hence it will be diffused out into the atmosphere. But it is not a waste. It is not the waste product because all other organisms required it for their existence, including plants, because plants do require oxygen during night as well. The food manufactured in the leaf is required for the use by all other parts of the plant. So the glucose which is formed in the leaf, in either form, either sucrose or starch, it will be transported to the other parts of the leaf wherever it is required on priority basis. If the flowering season is going or a fruit season is experienced by the plant, the food is more transported or translocated towards that part of the plant, or simply it will be stored in the form of starch, either in the stem or in the root. There are certain factors which affect the photosynthesis. Number one is external factors include light intensity and carbon dioxide concentration. Please remember these two factors, they are interlinked. We can say that the rate of photosynthesis will increase with the light intensity. To some extent it is true, as you can see in this picture. Photosynthesis will increase with the light intensity up to the certain limit only. And when it gets stabilized, that time the concentration of carbon dioxide will be 0.02%. But if due to some reason at this point the carbon dioxide concentration increases, the photosynthesis will also increase again up to the certain limit or up to the certain concentration of carbon dioxide, that is 0.05%. Over and above, if the carbon dioxide concentration increase, the rate of photosynthesis will not increase because stomata it will get closed due to increased CO2 concentration. Third factor is temperature. With the rise in temperature, rate of photosynthesis also rises. This rise can occur up to maximum 35 degrees Celsius, after which the rate will fall down and the process can be stopped completely at 40 degrees Celsius. Because at this temperature, the enzymes present inside the plant cell will get destroyed. Similarly, if the temperature decreases, the rate of photosynthesis also decreases, but we can see that there is a doubling of the rate of photosynthesis in rise from temperature in every 10 degrees Celsius. For example, 20 degree to 30 degree Celsius, if the temperature is the temperature range exists, that time suppose 40% photosynthesis is taking place. But if the temperature rises from 30 degree to 35 degree, because 40% we can't take, the process will get stopped. So up to 35 degrees Celsius, if we can see, then the rate of photosynthesis can become double from 40% to 80%. Fourth factor is water content. The scarcity of water due to two important reasons. Rate of absorption will be reduced or the rate of transpiration might be increased. Both of these will responsible for reduction in the rate of photosynthesis because stomata it will get closed. Always remember, only 1% of the water absorbed by the root, it is utilized in photosynthesis. Remaining entire water, it will get transpired. Internal factors responsible for the photosynthesis will be chlorophyll, protoplasm, and structure of leaf. Chlorophyll, as you know, it contains nitrogen, magnesium, which will serve as the most important minerals for the functioning of chlorophyll, and its deficiency can cause the drop in trapping of solar energy, hence the rate of photosynthesis. Protoplasm, dehydration of protoplasm for some reason, that is plasmolysis, as we studied in lesson number 4, reduces the rate of photosynthesis. Similarly, if more amount of sugar and starch it get accumulated inside the cell, that also reduces the rate of photosynthesis. Structure of leaf may include the thickness of cuticle, uneven distribution of stomata, surface area of the leaf. All these factors can influence the amount of light and the amount of carbon dioxide entering in the leaf. So hence we can say these are the factors which are controlling the process of photosynthesis. Experiment on photosynthesis. The first step which is required to perform any experiment based on photosynthesis is destarching. That is called as removal of the starch. A plant used for experiment on photosynthesis need to be destarched by keeping it in a dark room for 24 to 48 hours. During this period, what will happen? All the starch, it will get removed from the leaf, because the plant cells or the plant organs, it will utilize that starch for the energy purpose. Now the destarched plant is defined as it is a one whose leaves are free from starch. How we can say, or how we can confirm that whether the leaf or the plant is completely destarched? We have to follow these 4 steps. Step number 1, dip the leaf in a boiling water for a minute so that it will kill the cells. Later on, we have to boil the leaf in a methylated spirit or methanol, or if it is not available, ethanol also can do, in a water bath, so that all the chlorophyll which is present inside the plant cells, it will get removed. Because of this process, the leaf will become hard and brittle. Therefore, we will keep it in a lukewarm water so that the leaf will get softened. Later on, we have to spread it in a petri dish and we have to pour iodine solution. The presence of starch can be identified by the blue-black color, which indicates the presence of starch if it is present. If the starch is absent, the color of the leaf will become brown. Experiment number 1, to show that chlorophyll is necessary for photosynthesis. For that, we have to take a plant with a variegated leaves. Variegated leaf means a leaf is having some green and some non-green areas. For example, coleus, croton, and geranium. Destarch the leaf by keeping it in a plant, by keeping the plant in the dark room for few days. Place the plant in the sun. After few hours, you have to pluck one leaf as shown in the figure. Make its outline on a paper and mark the green and non-green areas. Right? Test the leaf for the starch. Only the green color part of the leaf, it will turn bluish black, showing the presence of starch. Second experiment, to show that sunlight is necessary for the photosynthesis. For this, we will take a plant with destarched leaf, cover one of the leaf with the black paper on which a design is cut. You can cut whichever design you want. Place this plant in the sun and after few hours, test the leaf which is covered by the black paper for the presence of starch. We can observe that only the part of the leaf which could get light convert, or it will shows the bluish black color area. The part which was covered by the black paper, it will turn brown because there was no photosynthesis take place. Third experiment, to show that carbon dioxide is necessary. Take a plant with the discharged leaf, insert the plant half of one of its leaf into the conical flask, which must contains potassium hydroxide. which will serve as an absorbent for carbon dioxide. Leave the plant in sunlight, and after a few hours when you test this leaf, correct, for the starch, you will see that there is no starch is produced. The leaf will become bluish, sorry, the leaf will turn brown, correct, because all the carbon dioxide present in the conical flask absorbed by the KOH, so there was no source of carbon dioxide present. Photosynthesis did not happen or occur. Experiment number 4, to show that oxygen is produced during photosynthesis. For that, we will require some aquatic plant, Alodia or Hydrilla or eelgrass or pondweed also can do for that matter. We will place this plant in a beaker containing pond water, or if the pond water is not available, we have to add some carbon dioxide source, that is calcium carbonate powder will do or will suffice the purpose. Invert, we have to place this plant in a beaker. Later on, we have to cover it with a short stem funnel, as you can see in this picture. Invert the test tube, which is full of water, over the stem of the funnel. We have to ensure that the level of the water inside the beaker is above the stem of the inverted funnel. Place this apparatus in the sunlight for a few hours. The bubbles of the gas which will be collected in the test tube later can be tested for the gas. The glowing splinter bursts into the flame, which shows the presence of oxygen. Carbon cycle is a series of the chemical reactions in which carbon as a chemical element, that is in its oxide form, that is carbon dioxide, is removed from the air used by the living organism in their body processes, and it is finally returned to the air. The essential steps, as you can see, CO2 will be taken up by the green plant through a process called as photosynthesis. Then it will become the part of the food chain. The green plants will be consumed by the consumers, that is animals. Both plants and animals, when they respire, they will give out the carbon dioxide. When green plants and consumers, the dead remains of these plants and animals, they are consumed by the bacteria and fungi. We call them as decomposers. And in this process, again the carbon dioxide gas is released. When fuel such as wood or fossil fuel like coal, petroleum, or the natural gas is burnt, the carbon contained in its oxidized state, that is carbon dioxide, is also released. Certain amount of carbon dioxide, it is also released during the heating or the combustion of limestone in lime kilns. This is all about the lesson photosynthesis, student. Thank you so much.